Genome-wide association analysis is a powerful tool for discovering genes associated with a trait, but misses many of them. PAST finds more of these genes and better completes the picture. PAST takes GWAS output and adds information about pathways that cells use to build metabolites and to create phenotypes in an organism. Genes in these pathways are often missed by GWAS alone.
After verifying that the GWAS data is tab delimited, use the Association File box to select an association file and the Effects File selection box to select an effects file. To load GWAS data with PAST in the R console, modify and run the code as indicated. To load linkage disequilibrium data with PAST Shiny, after verifying that the linkage disequilibrium data is tab delimited and contains the appropriate types of data, select the file containing the linkage disequilibrium data. To load linkage disequilibrium data with PAST in the R console, modify and run the code as indicated. To assign SNPs to genes with PAST Shiny, locating annotations in general feature format, select the file containing the general feature format annotations and modify the settings according to the window size and R² cutoff that are most suitable for the species being considered as necessary. To assign SNPs to genes with PAST in the R console, modify and run the code as indicated.